Genstat makes it easy to analyse censored Poisson count data using a log linear model, a generalized linear mixed model, or a hierarchical generalized linear model. During data collection, censoring occurs when counts cannot or are not taken above or below a particular bound. For example, if your experiment generates some very large counts, it may only be practical to count up to a certain value. Observations exceeding this value are therefore right-censored, as in this example where the number of insects is only counted up to 100. Genstat readily handles such censored Poisson data, providing you with well-behaved and reliable estimates of the model parameters and treatment means. Let's work through an example using one of Genstat's example data sets. This data was collected to test whether a ryegrass endophyte, in this case a specific fungus that lives within the plant, provides protection against a common insect pest. Better protected plants grow larger and produce a higher number of tillers. 2 different ryegrass cultivars were tested. The response variable of interest to the scientists was the number of tillers on the ryegrass plants. However, counting ryegrass tillers is time-consuming, so when they reached 200 tillers, they stopped counting and simply noted that the ryegrass plant had at least 200 tillers. Consequently, the observed tiller count data is right-censored at 200. As the experiment had a randomized complete block design, we'll analyze this data using a generalized linear mixed model, allowing for censoring. If we preferred, we could analyze this data using a log linear model, that is, a generalized linear model with a Poisson distribution and a log link, or using a Poisson hierarchical generalized linear model. We'll start by opening the generalized linear mixed model menu. The response variable is tillers. The fixed model, our treatments, is cultivar by endophyte by insect. The random model is replicate, reflecting the randomized block design. And the distribution is Poisson with a log link function. You'll recall that the scientists stopped counting tillers when they reached 200, so select the checkbox for counts and enter a censoring value of 200. The direction radio button states whether the counts are left or right censored. In our example, tiller counts were not taken above 200, therefore our data are right censored. Now click Options. In the Display section, we select which items of output we want to display on screen. Let's output a description of the model, the variance components, the predicted means on both the scale of the linear predictor and on the count scale, and also the walled tests. As we're analysing censored Poisson counts, we also have the option of printing the estimated values of the censored observations. Let's also display summaries of the standard errors of means and differences between the means, as well as a plot of the residuals. The other options control various aspects of the method used to fit the model, including the EM algorithm used to estimate values for the censored observations. We'll leave the other settings at their defaults, but remember, if you need to know more about any of these settings, just click the Help button. Click OK to close this dialog, then Run to perform the analysis. Here's our output, showing estimates of the censored observations and estimates of the treatment means, which take into account the censoring. The walled tests indicate the presence of a significant endophyte by insect interaction. To explore this, let's plot the means. Although the walled tests indicated very little difference between the 2 cultivars, we'll plot the full table of cultivar by endophyte by insect means, with least significant differences at the 5% level. Note that these means are plotted on the scale of the linear predictor, that is, on the log scale. The graphs clearly show the negative effects from insects on ryegrass plants that are not infected with the endophyte. There are fewer tillers on plants without endophytes. Conversely, when ryegrass plants are infected with an endophyte, the presence of insects does not reduce the number of tillers. Therefore, the data show us that for both ryegrass cultivars, the endophyte does provide protection against the insect pest. You can analyze censored Poisson data using the log linear modeling option on the Generalized Linear Models menu. You can also use the Generalized Linear Mixed Models menu or the Hierarchical Generalized Linear Models menu.